Welcome to the Deep Dive, where we wade through a stack of research news and notes so you don't have to.
That's right.
This time we're taking a deep dive into a world that's both incredibly complex and absolutely essential to modern biology, the world of algorithms.
Yeah.
Specifically, algorithms designed to untangle the mysteries hidden within our DNA.
That's right. We're talking about the tools that help us make sense of massive amounts of genomic data. Those long strings of A's, T's, C's, and G's that hold the blueprint of life.
Okay, I'll admit, just hearing the word algorithm makes me think of complicated code and equations.
Right.
But I have a feeling you're going to make this fascinating, right?
Absolutely. Think of it like this. You have a giant puzzle, a jigsaw puzzle, with billions of pieces.
Okay.
And each piece is a tiny fragment of DNA. Algorithms are the tools that help us put this puzzle together, revealing the bigger picture of how our genes work.
All right, it's a pretty good analogy.
Yeah.
So where do we even begin with this genomic jigsaw puzzle?
Well, one of the fundamental challenges in genomics is searching for specific sequences within vast amounts of DNA data. Imagine you're trying to find a particular sentence in a library full of books.
Right.
You need a way to search efficiently, and that's where algorithms come in.
So it's like having a super-powered search engine for DNA.
Exactly. And one of the early tools developed for this purpose is called a suffix array. It's like creating a highly organized index for a text listing all possible suffixes, basically every substring from a given position to the end in alphabetical order.
So instead of reading through the entire DNA sequence like a giant book, we can use this suffix array to quickly jump to the sections we're interested in.
Precisely.
That's
It's like
cool.
using the index of a book to find specific topics.
All right.
And because it's sorted alphabetically, you can use a very efficient search technique called binary search, which is incredibly fast even for massive data sets.
That makes sense.
Yeah.
But I imagine there are limitations, right?
Yeah.
No algorithm is perfect, and we're talking about enormous amounts of data here.
You're absolutely right. While suffix arrays are great for finding exact matches, they can be quite memory intensive. The larger the text, the bigger the suffix array, and the more memory it consumes.
So there's a trade-off between speed and memory usage.
Yeah.
It's like choosing between a sports car that's fast but has a small trunk and a minivan that's spacious but not as quick.
That's a great analogy.
Yeah.
And researchers have developed various techniques to optimize suffix arrays, making them more efficient for specific tasks. For example, there's something called a spaced suffix array.
Okay, a spaced suffix array, what does that mean?
Imagine you're searching for a specific pattern, but you don't need to match every single letter. A spaced suffix array allows you to focus on key positions within the pattern, like looking for specific landmarks on a map.
Okay.
This can significantly speed up the search, especially when dealing with similar but not identical sequences, which is often the case in genomics.
So it's like using a filter to highlight the most important parts of the sequence, making the search more targeted and efficient. I like it. But I have a feeling there are even more tricks up their sleeves when it comes to analyzing DNA.
You're absolutely right. There's another fascinating tool in the genomic analyst's toolbox, Bloom filters.
Okay.
They're a bit like those yes-no oracles, quickly telling us if a specific element is likely present in a data set without having to search through every single item.
Okay, that sounds intriguing.
Yeah.
So instead of combing through the entire DNA sequence, we can ask this Bloom filter, hey, is this specific sequence here? And it gives us a quick answer.
Exactly. And what's remarkable is that they do this with incredible efficiency, even for massive data sets. They're particularly useful in scenarios where we need to quickly check for the presence of specific sequences, like identifying potential matches before running a more detailed analysis.
So it's like a quick pre-screening tool, helping us narrow down the possibilities before we dive into the nitty-gritty details.
Right.
It's all about working smarter, not harder.
Precisely. And while Bloom filters are remarkably fast, they do have a small chance of giving a false positive, telling us something is there when it's actually not. But this trade-off is often negligible compared to the massive speed gains they offer.
I see. It's like having a security guard who might occasionally let someone in by mistake,
Yeah,
but overall does a fantastic job of keeping things secure.
that's a great way to put it.
Yeah.
And when it comes to genomic analysis, accuracy is paramount. So researchers have developed specialized Bloom filters designed specifically for DNA sequences called camera Bloom filters.
Okay.
These KBFs leverage the inherent overlap between cameras and a sequence short stretches of K letters to significantly reduce those false positive rates and minimize memory usage.
So it's like having a security guard who knows the entire guest list by heart and can spot imposters with even greater accuracy. But with all these fascinating algorithms,
Yeah.
where do those MEMS algorithms, the stars of our deep dive, fit into this picture?
Right.
Are they related to these suffix arrays and Bloom filters?
They are indeed. MEMS algorithms, which stand for Maximal Exact Matches, take a slightly different approach, but build upon some of the principles we've discussed.
Okay, I'm all ears. I'm ready to dive deeper into the world of MEMS and see how they stack up against these other approaches.
Well, let's shift our focus to MEMS algorithms specifically. Instead of just finding any match, MEMS algorithms are like those meticulous detectives we see in movies, focusing on finding the longest possible exact matches between sequences. They're the masters of pinpointing those crucial connections within the vast sea of genomic data.
All right, that sounds pretty impressive. But before we get lost in the details, why are these maximal exact matches so important? What can they tell us about our DNA?
They're crucial for a wide range of applications, from comparing genomes to identify evolutionary relationships to pinpointing disease-causing mutations. Think of it this way. Imagine you have two versions of a book, one original and one with some edits.
Okay.
By finding the longest identical passages, you can quickly identify the sections that have changed, revealing the key differences between the two versions.
That's a brilliant way to put it.
Yeah,
So MEMS algorithms are like those highlighters we use to mark up important text, except they're highlighting the most significant similarities and differences within our DNA.
exactly. And in the next part of our deep dive, we'll unravel the inner workings of these MEMS algorithms, explore the clever tricks they use to achieve remarkable efficiency, and discover how they're shaping the future of genomic research.
Well, you've definitely piqued my curiosity. I can't wait to see how these MEMS algorithms stack up against those other approaches we've discussed. Join us in part 2 as we continue our deep dive into the world of algorithms and their impact on understanding the code of life. Welcome back to the deep dive. In part 1, we explored some fascinating algorithms like suffix arrays and Bloom filters, which are all about finding those needles in the haystack of genomic data. Now we're focusing on the stars of the show, MEMS algorithms. They're not just looking for any match, they're seeking out the longest possible exact matches, those crucial connections within our DNA.
That's right. And what makes MEMS algorithms so compelling is that they can do this with incredible efficiency even when faced with massive data sets.
Okay, that sounds impressive, but I have to admit I'm still a bit fuzzy on how these algorithms actually work. It's like they have this magic ability to sift through billions of letters of DNA and pinpoint those longest exact matches. How do they do it?
It's not magic, but it is incredibly clever. One way to think about it is like this. Imagine you're comparing two long strings of beads, each bead representing a letter in a DNA sequence. You start by lining them up and looking for matching beads.
Okay, I can picture that. So as we move along the strings, we keep track of the longest stretch of matching beads we've found so far. But how do we know when we've found a maximal exact match? The absolute longest possible match.
That's where the algorithm's logic comes in. It maintains a set of invariance conditions that always hold true as it scans through the data. One key invariant is that it won't report a match that ends before a certain point unless it's already found all the matches ending earlier.
Wait, can you give me an example? My brain is starting to get a bit twisted.
Absolutely. Let's say we're looking at the sequences ACTG, GCTA, and GCTG. As the algorithm scans, it might first find the match, ACTG, but it won't immediately report this as a mem. It keeps going and finds ACTGG, which is longer. Only then does it report ACTGG as a mem because it knows there can't be any longer matches ending before that point.
Ah, I see. So it's like a meticulous detective carefully gathering all the evidence before declaring a case closed.
Right.
It ensures that no potential match, especially the longest ones, slips through the cracks.
Exactly. And as the algorithm progresses, it keeps track of what we call active positions. These are points in the pattern, or GCTG in this case, where there's a potential ongoing match with the text.
Yeah.
For each active position, it remembers the longest suffix of the pattern that matches the text up to that point.
So it's like the algorithm is juggling multiple possibilities at once, keeping track of all the potential matches as it moves along the sequences.
Precisely. It's constantly updating the set of active positions based on both matches and mismatches, discarding those that lead to dead ends and focusing on the most promising leads. This makes the process incredibly efficient, minimizing both time and memory usage.
Okay, I'm starting to get a real feel for how these MEMS algorithms operate. They're like those super sleuths who can quickly eliminate suspects and zero in on the key evidence. But I remember you mentioned earlier that they face challenges when dealing with the sheer volume of genomic data.
That's right. Even with clever strategies.